ZUMA-1 trial is testing a CAR T-cell approach. So most people will know that CAR is chimeric antigen receptor T-cells. So these are T-cells taken from the patient and transfected with a receptor that will bind to CD19. And the ZUMA-1 trial was reported out previously with very encouraging response rates in very refractory patients. So this study specifically looked at patients with primary refractory or very heavily pretreated and refractory large cell lymphoma patients. And in that subset of patients, the response rate was high. We've seen response rates around 60–70% with durable responses in complete responses seen in about 40% of patients. The long-term follow-up, which was now presented with follow-up extending out to a year, the key question was just, are these responses durable? And that's, I think, what was very encouraging about the ZUMA-1 longer-term follow-up, is that it does seem to be the patients that have responded, had a complete response, are remaining in remission, and the long-term follow-up looks quite promising. I think I know there are studies currently ongoing that are looking specifically at comparing this now to standard approaches such as autologous stem cell transplant. And I think that's going to be the very exciting next direction for the field, because if it turns out that CAR T cells are superior to utilizing the standard autologous stem cell transplant, that's going to be a huge move forward in the field. But the other thing that I think has made practical changes that really are exciting for patients and exciting for physicians treating patients is that CAR T cells produced as per the ZUMA trial actually are now approved therapies which are being utilized in practice as well.
CAR T cell is one of the more exciting and intensive therapies in diffuse large B-cell lymphoma in terms of not only efficacy but toxicity profile. And it will be important moving forward that as these treatments are brought to the public, that institutions with experience of not only the procedure but the side effects are utilized initially in the treatment of patients with this disease. There are few treatments that have shown as much promise as CAR T cell in hematologic malignancies. That being said, we need to continue to evaluate relapses moving forward before anyone would say that these are curative measures in lymphoma at this time. There are a variety of different CAR T platforms that are being put forward by multiple companies and even institutions at this time. And this is an evolving treatment. Identifying which of these therapies provides the most benefit with minimal or at least limiting toxicity profiles is going to be important. The JCAR study looked at a different company's product and may demonstrate differences in T-cell subpopulations in terms of benefit in these settings. However, I think ultimately we need further testing and further refinement of CAR T-cell before we can truly identify the best platform to use in lymphoma. And I think that while we have a number of different companies evaluating this, the answer to that question is to be determined in the future.